All right, this mini lecture is about the citric acid cycle, which takes place in the mitochondrial matrix. And so all these components are floating in the mitochondrial matrix, and there are 8 steps, 8 chemical reactions to the citric acid cycle, and each of these has a specific enzyme. And so the starting reactants, the substrates, in this case are citrate, and the product is isocitrate. And so each of these is a chemical reaction, and we don't need to know the names of the enzyme. We don't need to know the names of the substrate or the structure of these. And so the citric acid cycle is very dense, but we need to take out from it just a few things. One is that it is happening here, that these are chemical reactions like we're learning about, that they are catalyzed in each case by a specific enzyme and protein enzyme. They're all floating in the mitochondrial matrix. And then each of these steps is exergonic energy releasing, and so they all would have a negative delta G, so this process is spontaneous, it's tumbling downhill. And some of the steps are larger than other steps. And when the step is large enough, ATP or NADH or FADH are captured. So see, they're all in yellow there. Those are the energy yielding steps. So here's a good example where we have the substrate isocitrate, and it is converted to alpha ketoglutarate. So that's the sugar, the carbon's starting to get oxidized. And here, one of the products is CO2. And you'll see when CO2 is released, that tends to be a very energy yielding step. There's an increase in entropy that's happening there. We're actually releasing, then simplifying that molecule that we're using. And anytime there is a big energy drop, that is going to be used to catalyze the synthesis of one of these energy intermediates. And we've talked about that reducing NADH takes a lot of energy. So we can just go through this logic and for just one of these, and we can say, okay, so ATP requires a plus 7.3 kilocalories per mole to synthesize. NADH in oxidative phosphorylation, the third step of cellular respiration, NADH is going to be worth 3 ATP. So it must be worth at least 3 times 7.3 kilocalories per mole, or 21.9. And we've said for reactions to be spontaneous and forward moving, we need to have, so those are our 3 ATP, we need to have this NADH drop to be greater than the energy it's going to drive. And so if this is the case, if one NADH can make 3 ATP, then this energy drop must be greater than minus 21.9. So that when we add them together, we still have a negative delta G and a spontaneous reaction. Okay, so if this is greater than negative delta 21.9, then this reaction where we're making the NADH, we're pushing NAD plus uphill to reduce it to NADH, that must have a greater than plus 21.9 kilocalories per mole, right? Because if it's going to be this much down, it's going to be that much up. And because of that, from isocitrate to alpha ketoglutarate, we must be dropping more than 21.9 kilocalories per mole. So there's just a simple logic, and it's not tricky at all. It's just sensible logic of you can't get something for nothing. If you want to build 3 ATP during oxidative phosphorylation, and we'll do that in our next mini lecture, if you want to build 3 ATP, you're going to have to give up enough energy to do that, and that's how much energy it's going to take, so it's going to have to be more than that. If that's how much energy is released, it's going to take that much energy to build it in the first place. You're going to have to release that much energy from the sugar that you are oxidizing and metabolizing here. So in just that one step, it's tremendously exergonic, and we can couple it to this synthesis. This, we'll learn over here, just as a second example, that FATH is equal to 2 ATP in oxidative phosphorylation. So what's going on? What's going on in the conversion of succinate to fumarate? And the answer is that this enzyme is able to catalyze that, release that energy, so it helps get it up over that activation energy barrier by providing an alternative transition state, like we learned about a couple mini lectures ago. So this enzyme helps this reaction occur, and when that energy is released, it's coupled to the synthesis, and obviously this reaction can't be releasing enough to make NADH, otherwise it would, trying to get as much energy as you can out of it. It must be releasing something less than this, so that we can't capture an NADH, but we could capture some FADH. Then what's going on here is, well, there's some ATP coming out here. It's actually coming out as GPP and then getting converted to ATP. But this one, succinyl-CoA to succinate, must give up even less energy, right? What about over here, fumarate to malate? Well, apparently it's downhill because it's happening spontaneously, but it's not enough to make ATP. So this downhill drop would be less than minus 7.3 kilocalories per mole. Why does it occur? Because these chemical reactions that don't provide energy are setting up the other chemical reactions so that we have a sort of an overall setup and then the places where we capture energy. And that's all we really need to say about the citric acid cycle. This is where carbon dioxide is peeling off, and so we're going to knock this sugar all the way down to nothing. We're completely converting the carbons and releasing them. Here's a nice flow chart of our overall energy diagram, where this is the free energy, and this is the progress of the reaction. And you can see that in glycolysis, there's actually an energy investment phase where it takes some ATP to do the setup, and then there's a big energy drop, makes some NADH during glycolysis. There's an energy drop to make some ATP. Here's where we are getting the setup to get it into the mitochondria. We get some more energy out there. And then here's a couple of the big drops where we get C, and they're all relative sizes. So what we want to focus on in the citric acid cycle is these energy releasing steps and the logic of the energy. That's all there is to that. So we're doing great. And then here's our first look at the final summary figure from chapter 9, and chapter 8, and that is the amount of ATP released. So in glycolysis and the citric acid cycle, there's a little bit of ATP produced directly, and it's produced by a process called substrate level phosphorylation, and we need to know that. And substrate level phosphorylation is where the enzyme that catalyzes those steps is able to catalyze. It's almost like a teeter-totter, where one enzyme has two active sites, and one active site binds the sugar, and the other active site binds the ADP. And it tips back and forth like that. It actually physically moves in order to connect the energy release, bending of the protein molecule, the energy released of one chemical reaction of the sugar digestion to the actual synthesis of ATP. So a pretty amazing enzyme that's present in our body, inside the mitochondria, or in the cytoplasm or mitochondria, that is able to directly convert energy into ATP through that chemical reaction. Then the vast majority of the ATP is produced during oxidative phosphorylation, and that is energy that was captured in glycolysis and the citric acid cycle in the form of NADH and FADH2, and those are just getting converted in the third part of the cellular respiration. And so that's all we want to say about citric acid cycle. So there's some really important things to learn about it, but limited